Hi students, today we are going to start a very important topic which is PCR. It stands for polymerase, means many polymerase. P for chain and R means react. We can say that a chemical reaction which is going to happen many times. That means poly word is used, means repetition. Repetition. So polymerase chain reaction. In short, we can say that PCR. So this technique, it is a technique or molecular biological technique which was invented by a scientist, Carrie Mullis, Carrie Mullis, in 1983. So in 1993, Carrie Mullis got Nobel Prize for this discovery of the technique for the PCR and in the field of for the chemistry. So PCR, it is a method which is performed by a machine, and machine is called as thermocycler. Thermo means heat, means it is a technique which use heat for the amplification of desired product, or we can say that amplification of the DNA molecule. It is a genetic material, and in this PCR, in this method, we use the DNA for the amplification of the desired product, or we can say that a specific reason of the DNA molecule without the requirement for time consuming cloning procedure. So PCR reaction takes place in the Eppendorf tube. Eppendorf, E-double-P-E-N, Eppendorf tube. It is a plastic tube and the structure is conical in shape. So in this tube, we make a master mix of the different type of chemical and plus our desired DNA fragment. So using PCR technique in very very low amount or the quantity of the DNA content available from sample of different samples like blood, semen, saliva, or even minute particles like skin fragments and many things, hair, scalp, etc. can be amplified many times and also analyzed. In this technique, a very important, or we can say that backbone of this technique, which is Taq polymerase enzyme Taq polymerase enzyme is used. Taq means thermos and AQ means aquaticus. Aquaticus, Thermus aquaticus, it is a bacteria which is found in heat, or we can say that thermal vents, hot thermal vents, which is a temperature of more than 90 to 100 degrees Celsius, so that this bacteria is resistant on this temperature. That's why we use Taq polymerase enzyme in this technique. Also, Taq polymerase enzyme used in PCR, which is a special type of DNA polymerase enzyme, which is resistant to high temperature. Also, another type of enzyme, that is PFU, is used. PFU, isolated from Pyrococcus furiosus bacterium, and vent polymerase, that is isolated from Thermus litoralis bacteria. So mainly three steps, three steps followed in the PCR machine or thermocycler machine. First one, that is denaturation. Denaturation. Second one, that is annealing. And the last one, that is the third one, which is extension. These three procedure or three steps followed in this reaction. So first one, that is denaturation. In this process, our DNA, or we can say that double-stranded DNA molecule is placed at the 94 degrees Celsius. So double-stranded molecule is getting denatured. Suppose that this is our double-stranded molecule. In this temperature, they isolated from each other and converted into single-stranded, and each single-stranded function as a template molecule. Second one, that is annealing. In this temperature of this machine, 54 degrees Celsius. In this step, two primer is used. Two primer DNA are attached on the 3' end, 3' end of the single-stranded DNA, and the last one, that is extension, that is 72 degrees Celsius. On this temperature, our enzyme, that is tacpol, in short, enzyme synthesize DNA strain over the template. DNA is automatic process because of Taq polymerase enzyme is heat resistant. So through this process, we can amplify our desired small quantity of the DNA molecule. Now, finally, the application of this procedure, where is this used? PCR technique. So the first one, in recent studies, this is used in the different type of research processes or research experiment, or we can say that research institute also. Next one, in COVID, we know that this viral infection is also the identification identified by this PCR technique also, and different type of disease detection of the disease through this PCR technique also, and different type of pathogenic microorganisms, because PCR gives the accurate result accurate result with the very small amount or small quantity of that DNA fragment. Also, DNA analysis for the archaeological studies, we can use the PCR also. So there are different type of places in which PCR is used. So few examples of this related to the application. Hope this answered your question, or this topic is understandable. Thank you, students. Thank you.